As human beings adopted a settled lifestyle, they cultivated different grasses to develop cereals, such as wheat, rye, maize, and barley. Crossing different plants permitted an empirical increase in yield, thereby improving the human food supply. Gregor Mendel opened up new, systematic methods of modifying plants genetically, thus making a wide variety of food sources accessible. This also led to an increase in the amounts of valuable nutrients in the food chain, and thus the quality of nutrition. With technical progress, civilization has taken another step forward. As a result of medicine and its successes, the average age increased in the 19th and 20th centuries, and acute and preventative measures brought about decisive improvements in health and the quality of life. The extremely rapid development of molecular biology gave rise to the first generation of human therapeutic proteins for substitution therapy in medicine. The classical products are insulin, human growth hormone, interferon alpha, tissue plasminogen activator, and erythropoietin. It is now possible to adapt these biopharmaceuticals by specific modifications to therapeutic requirements. The fine regulation of intact genes and the proteins formed from these is responsible for maintaining the physiological status of the human body. The correct nucleotide sequence is required to ensure the correct amino acid sequence in the protein molecule. The substitution of nucleotides can lead to a change in the amino acid sequence and a non-functional protein molecule, and thus to metabolic disorders. Genetic engineering permits the isolation of the genetic information for clinically relevant proteins produced in the body. Biotechnology makes use of microorganisms or cell cultures to produce these protein molecules in their native form. This has become possible in recent decades as a result of innovative manufacturing procedures and investments in production facilities. Substitution therapy can compensate for genetic defects. It can keep people alive, and it can improve the quality of life. Between 1982 and 2005, the first generation of biopharmaceuticals enabled enormous progress to be achieved in therapy. Biopharmaceuticals replace the defective physiological molecule. For example, during fibrinolysis, plasminogen attaches itself to the fibrin strand and is activated by tissue plasminogen activator. This leads to lysis of the thrombus. With the current wider-ranging possibilities of genetic engineering, natural molecules can be better adapted to the therapeutic requirements. The point mutation in position 103 means that tissue plasminogen activator is glycosylated at that point. The point mutation at position 117 means that the sugar residue with the high proportion of mannose is absent at that point. These structural changes minimize the binding of the molecule to liver receptors. This leads to a substantial increase in the half-life from 3 to about 30 minutes. The amino acid sequence 296 to 299, lysine, histidine, and 2 arginines has been replaced by 4 alanines. PAI-1 resistance is increased and fibrin specificity improved, while the fibrinogen level is maintained. The minimal changes to the native structure in the TNK-TPA molecule explain the absence of any substantial antigenic potential. TNK-TPA is the only single bolus thrombolytic that offers patients the earliest possible treatment. At the Vienna facility, immunology and oncology research has made crucial advances in the use of immunotherapy with monoclonal antibodies through the neutralization of antigens and tumor receptors, albeit at high therapeutic doses. Monoclonal antibodies permit, for high-potency cytotoxins, specific targeting of the toxin to tumor cells, thereby reducing the treatment dose. The immunotoxin conjugate is internalized by the tumor receptor. The intracellular pH cleaves the toxin from the antibody and expresses its anti-tumor activity locally at this point. With the aid of genetic engineering, it has been possible to develop antibodies for the neutralization of tumor necrosis factor alpha. Because an antibody, in principle, only neutralizes a single target, high doses are often required in therapy in order to neutralize large numbers of targets. At the Biocampus Cologne, Derevo Biotech AG is carrying out research into the irreversible catalytic inactivation of therapeutic targets using proteases with completely new tailor-made specificities. The protein scaffold chosen was human trypsin with a broad substrate specificity. By introducing randomized loops into the protein scaffold, it is possible to generate a large number of highly specific proteases with different target specificities. With ultra-high throughput screening systems, suitable proteases for the inactivation of pharmaceutically relevant targets, such as tumor necrosis factor alpha, can be selected without inactivating other serum proteins. This mechanism of action has the advantage over antibody immunoaffinity that many tumor necrosis factor alpha molecules can be cleaved catalytically by a single protease molecule. The pro-inflammatory activity of tumor necrosis factor alpha in inflammatory cells is effectively suppressed by the catalytic degradation of tumor necrosis factor alpha. These microbially produced trypsin proteases thus offer an alternative to monoclonal antibodies as a possibly superior treatment for immunological diseases. Skill Proteins GmbH in the Halle Science and Innovation Park is engaged in the generation of artificial binding proteins. The protein scaffold chosen for the production of so-called aphelin molecules is gamma crystallin. This is a small and extremely stable structural protein from the lens of the eye, where its purpose is to refract light. By making targeted changes in the amino acid sequence in the beta-pleated sheet while the tertiary structure is maintained, the protein scaffold can be given high affinity for selected targets. In this way, high-affinity binding proteins with low dissociation rates can be produced. Because of their low molecular weight of only 20 kilodaltons and the absence of disulfide bridges, aphelins, unlike monoclonal antibodies, can also be used to neutralize intracellular targets. Aphelins can be applied in affinity chromatography. Replacing up to 8 amino acids in the aphelin beta-pleated sheet produces high affinity for monoclonal antibodies. The physico-chemical properties of aphelin proteins, such as pH and temperature stability, support its use in the purification of monoclonal antibodies. The C-terminal end of the aphelin molecule is coupled to cephalose by means of a thioether reaction without affecting the binding epitope. In collaboration with Boehringer Ingelheim, new affinity chromatography materials for the purification of monoclonal antibodies are under development using this technology. Pyrrhus Proteolab AG, a biotech startup company located at the Weihenstephan Research Campus near Munich, has developed the anti-kalin technology, providing a novel class of therapeutic proteins. Natural members of the so-called lipokalin protein family are present in almost all organisms, where they serve to ensure the transport of metabolic substances. They share a common beta-barrel structure, but show pronounced structural differences in a loop region that makes up the ligand pocket. Reshaping this binding site allows anti-kalins to be engineered. These can develop specific binding activities for a wide variety of target molecules. Siderokalin is a natural lipokalin that has already been used for therapeutic application. In the human body, it normally binds and neutralizes siderophores, certain iron transport compounds that are released by microorganisms. The loop region of this lipokalin was successfully reshaped to yield an antikalin with high affinity for the cell surface receptor CTLA-4, which is expressed on activated T cells. When CTLA-4 interacts with its counterreceptor B7.1 on antigen-presenting cells, the T cell is deactivated. This is part of a self-regulatory mechanism of the immune system. Antikalin, with its specific binding affinity for CTLA-4, blocks interaction with the counterreceptor, and thus the T cell remains active for longer. The resulting stimulatory effect on cellular immune reaction can be used for the treatment of tumors. Avidia Incorporated in Mountain View, California, is synthesizing genetically tailored avomers, which are made up of several 4.5 kilodalton domains of human serum proteins. Within a domain of 40 amino acids, which form the protein scaffold involving 3 disulfide bridges, up to 28 amino acids can be modified. This produces high specific affinity for various target molecules. Avomers are protease and temperature stable. The anti-interleukin-6 avomer is optimized for the inactivation of interleukin-6. Crohn's disease and rheumatoid arthritis are its intended indications. It is advantageous to use microorganisms to generate these protein scaffolds. As they are less expensive to produce, they offer a competitive alternative to monoclonal antibodies. Boehringer Ingelheim, Austria, has developed franchise technologies for protein scaffolds, which are available to our partners for contract development and production. Therapeutic proteins can be pegylated to increase the half-life after parenteral administration. In the liver, beta-ferron is metabolized with a half-life of 5 minutes. This means that daily parenteral administration is required. At the N-terminal end of the protein, beta-ferron can be specifically pegylated and thus protected from rapid degradation in the liver. A longer half-life can reduce the number of doses to weekly parenteral administration. Because of their protein structures, these biopharmaceuticals have so far been administered by the parenteral or subcutaneous route, as they are digested in the gastrointestinal tract when administered orally. Using natural transport systems for antibodies in lung epithelial cells allows for inhalation and active resorption of biopharmaceutical fusion proteins. Antibodies are transported by the bloodstream to the various organs. By pinocytosis, they are transported through the endothelial cells of the blood vessels protected by the neonatal Fc receptor in endosomes. By the same mechanism, antibodies are transported through the epithelial cells of the lung and released to the lumen of the lung, where they can neutralize antigens. This mechanism, however, is reversible. Antibodies can be taken up by the pulmonary epithelium by phagocytosis. The vesicles merge with endosomal vesicles which express neonatal Fc receptors that bind the Fc fragment of immunoglobulin G. This protects immunoglobulin G from proteolytic destruction. The vesicle transits the cell and immunoglobulin G is released into the blood vessels. In Waltham, Massachusetts, Syntonix Pharmaceuticals Incorporated uses this natural absorption mechanism for the delivery of therapeutic proteins. The Fc fragment of immunoglobulin G is conjugated genetically with monomeric erythropoietin. With the Fc part of the monoclonal antibody, the therapeutic protein is transported from the lumen of the lung through the epithelial cells of the lung and the endothelial cells of the blood vessels into the bloodstream. After inhalation, the erythropoietin Fc fusion proteins are deposited in the upper and major airways, as demonstrated by scintigraphy, where the specific absorption via neonatal Fc receptors leads to a bioavailability of 35% on average. This non-invasive administration provides a potential therapy for chronic diseases. Biocerentalk Incorporated in Kyoto develops innovative drug delivery technologies for oral administration of therapeutic peptides and proteins via a gastrointestinal mucoadhesive patch system called GMAPs. The system consists of an insoluble protection layer, which protects the drug from digestive enzymes. The drug layer contains an absorption enhancer. An adhesive layer is coated by a pH-sensitive surface layer that only dissolves at an alkaline pH. The GMAPs are administered via gelatin capsules. This protects the drug from acid degradation in the stomach. After oral administration, the gelatin capsule dissolves in the gastrointestinal tract. At the target site of the small intestine, the surface layer of the GMAPs dissolves and the adhesive layer adheres to the intestinal mucosa, allowing for optimal contact for absorption. Absorption enhancers in the drug layer optimize the absorption of the therapeutic peptides and proteins. The result is an average bioavailability of 11% for erythropoietin, 10% for insulin, and 8% for interferon alpha, an essential treatment for hepatitis C. This allows direct targeting of the liver, frequently the first site of biopharmaceutical activity. These second-wave technologies are at the focus of biopharmaceutical research and development at Boehringer Ingelheim. Second-generation molecules with enhanced efficacy and a prolonged half-life tailored to therapeutic needs. Protein scaffolds with catalytic activity for therapeutic targets. Neutralizing protein scaffolds such as anticalins, avomers, and aphelins. Slow-release formulations with improved convenience for patients. Fc fusion proteins for active adsorption after inhalation. GMAPs for oral formulations of therapeutic proteins targeting the liver. Altogether, new dimensions in biopharmaceuticals. In strategic cooperation with research organizations worldwide, Boehringer Ingelheim is continuously improving its own research. The experience with these technologies is available to our partners in biopharmaceutical contract development and production. In the treatment of acute and chronic diseases, to the benefit of patients, the second generation of biopharmaceuticals is thus opening up new horizons.